Hello again. This time our lesson is about microbial extracellular components. This lesson will focus on the structure and functions of the possible components that can be found outside the cell wall of bacterial cells. At the end of this lesson, you will be able to identify the components found outside the bacterial cell wall and describe the structures and functions of the structures that are found outside the bacterial cell wall. So the motivation question for this lesson is this. Are microorganisms capable of movement? Can they protect themselves from harsh environmental conditions? And so, I hope you will be able to answer this question after this lesson. Now, if you look into the structure of a typical prokaryotic cell, of course, in our previous lesson, we have already discussed the properties of a prokaryotic cell. We have already defined what is a prokaryotic cell. Now let us go deeper into the different structures that we can find in a prokaryotic cell. Okay. Now the components of a prokaryotic cell are organized into the following. So first are those structures that are found external to the cell wall. The second is the cell wall itself. And the third are the structures internal to the cell wall. So let's first talk about the structures external to the cell wall in this lesson. Among the possible structures external to the prokaryotic cell wall are the Glycocalyx, Flagella, eggshell filaments, Fimbriae, and the Pline. Glycocalyx are substances that are secreted on the surface of prokaryotes. The word glycocalyx means sugar coat. This is a general term that is used for substances that surround cells. The bacterial glycocalyx is a viscous or sticky gelatinous polymer that is external to the cell wall and is composed of polysaccharide, polypeptide, or both. It is made inside the cell and is secreted to the surface of the cell. Now there are two types of glycocalyx. So if the substance is organized and is firmly attached to the cell wall, then we are describing a capsule. Well, if the material, the substance is unorganized and only loosely attached to the cell wall, then the glycocalyx is described as a slime layer. Okay? So again, capsule is organized and firmly attached to the cell wall, while slime layers are those that are unorganized and are loosely attached. Now, if you want to see glycocalyx or a capsule under the microscope, usually what we do is to perform negative staining, wherein you put a stain to color the environment around the cell so that the clear area between the cell contents and the surroundings that forms a halo is the capsule. So capsules in certain species are important in contributing to bacterial virulence. So again, when we say virulence, we are referring to the degree to which a pathogen causes disease. So capsules, they often protect pathogenic bacteria. Again, when you say pathogenic, we are referring to bacteria or an organism that is causing a disease. So they can protect pathogenic bacteria from phagocytosis by the cells of the host. We know that usually when a foreign or a harmful bacteria enters our body, it is the phagocytotic cell, usually macrophages, and other type of cells that will eat and digest and kill this bacteria. But those that are containing capsules may tend to escape phagocytosis and could live inside those cells that has engulfed them. For example, Bacillus anthracis. So they produce a capsule made up of D-glutamic acid. Now, according to research, only encapsulated na those with capsule na Bacillus anthracis causes anthrax, it is speculated that the capsule may prevent it from being destroyed by phagocytosis. Now, glycocalyx are also important in the formation of biofilms by microorganisms. Okay, so biofilms are any group of microorganisms in which the cells stick to each other on a surface. So they adhere to each other on a surface. So these adherent cells are frequently embedded within a self-produced matrix, what we call the extracellular polymeric substance, or the EPS. So EPS is a glycocalyx that helps cells in a biofilm attached to their target environment and also to each other. EPS protects the cells within it. It facilitates communication among them and enables the cells to survive by attaching to various surfaces in their natural environment. So, in other words, the biofilm is actually a whole new environment if you compare it to the surrounding environment of the cell. So, how are biofilms produced? So, when a microorganism, bacterial cells are attached to a particular surface, they may produce the EPS that could attract another group of certain organism. We call them the secondary bacterial species that would also be part of the biofilm until other species of bacteria may also enter and join the biofilm. So later on, some may be detached and could later on form another biofilm in other surfaces. Now, another extracellular material found in bacterial cells are the flagella. So, flagella are long filamentous appendages that propel bacteria. So, those bacteria that lack flagella without these projections are called astricuous. But if a bacterial cell may have flagella, they may be peritrichous or polar. Peritrichous meaning that the flagella is distributed throughout the bacterial cells. While polar bacterial cells are those that have their flagella only at the opposite ends of the cell or at the poles. Now, if they are polar, they may be monotrichous, lophotrichous, or amphitrichous. So, monotrichous, these are those cells with single flagellum at one pole. Lophotrichous are those that contains a tuft. This is a tuft of flagella coming from one pole, or they could be amphitrichous. Those that have a flagella at both poles of the cell. Eggshell filaments are another extracellular structure. These are found in spirosheets, and these this is useful for motility of the bacterial cells. Spirosheets move by means of eggshell filaments or endoflagella. They are bundles of fibrils that arise at the ends of the cell beneath an outer sheath and spiral around the cell have similar structure to flagella. The rotation of filaments produces a movement of the outer sheath that propels the spirosheets in a spiral motion. See, this is similar to a way a corkscrew is moved through a cord. So, corkscrew motion probably enables to move effectively through body fluids. Okay, another cellular structure is called the Fimbriae. So, Fimbriae are hair-like appendages that are shorter, straighter, and thinner than flagella and are used for attachment and transfer of DNA farther than for motility. This is found in gram-negative bacteria. The Fimbriae, singular Fimbria, can also occur at the poles of the bacterial cell, or that can be evenly distributed over the entire surface of the cell. They have a tendency to adhere to each other and to surfaces. And they're also involved in forming biofilms and other aggregations on the surfaces of liquids, glass, and rocks. They can also help bacteria adhere to epithelial surfaces in the body. So there are, for example, E. coli, they have the Fimbriae so that they could be attached to a particular surface. So it's almost the same as the Amorsico, the weed that we have in the Philippines. Finally, there are the pili, singular pilos. These are usually longer than Fimbriae and number only 1 or 2 per cell. They are involved in motility and DNA transfer. So the twitching motility plus a pilus extend by the addition of subunits of pilin. It makes contact with the surface and then retracts as the pilin subunits are disassembled. For gliding motility, the smooth gliding of movement of mixed bacteria is caused by the pili, and some of these pili are actually involved in conjugation. So this is now the pilus here, now the exchange of genetic material by these two types of bacteria. So this process is called conjugation. Okay. Now, to summarize this lesson, you should be able to answer the following questions. What are the different structures found outside the cellular microbial cells? What are the functions of these structures? So I hope you're able to answer this one after this lesson. Okay. So if you have questions and some clarifications, you can contact me through my email, or you can key in your questions in our Facebook Messenger group chat. Thank you very much, and see you again in our next lesson.